Hello, in this video we will discuss about the cell death, the type of the cell death. So totally four type of the cell death we will discuss in this video. So what is cell death? The loss of cell function lead to death because if survive, the cell cause uncontrolled cell division, cause tumor and malignancy. So in this way, the basically the cell which that is the external and internal factor involved for the cell death due to the damage of the DNA or inside the mitochondria or other factors which that involve for the cell death. So the DNA damage, endoplasmic reticulum stress as well as hypoxia, which that is basically intrinsic pathway, but the external is the basically the some cytokines or other death relating factors which that will cause the death when bind with the receptor. So let's begin to understand the cell death type. So number one is the apoptosis and necrosis, pyroptosis, and necroptosis. Apoptosis is the programmed cell death. It is used to early development to eliminate the unwanted cells like fingers development during the early age of the fetus. So on the other hand, the necrosis is the some uncontrolled external factors, death of the body tissue by injury and the radiation, chemicals due to lack of blood supply will lead to cause the death of the cell is known as the necrosis due to the injury. But the pyroptosis and necroptosis is the different, but it is similar like necrosis, but the cell death for the pyroptosis that is triggered by pro-inflammatory signals, not injury, but and associated with inflammation. So the inflammatory cells, macrophage, bacterial patho-infection, which that is also natural, and in this way the macrophage, which that will be die after the complete their mission. While the necroptosis is that triggered by innate immune response, so that is by rupturing dead cells and releasing intracellular component caused by toll-like receptor 3 and 4 agonist and tumor necrotic factor as well as the T cell receptor is involved for the death of the cell is known as the necroptosis. So let's begin to understand one by one with detail little bit. So let's begin to understand the necrosis. Necrosis, for example, when tumor necrotic factor bind with a receptor. So in this way, here you can understand this is the TRAD and TRAF. 2 and 5 will combine together to phosphorylation, will lead to recruit the RIP1 and CLAP. This CLAP will ubiquitination to the RIP, and after this the necrotic factor kappa B stimulation that will after the gene transcription to translation. While in this case ubiquitination is lead to caspase 8 inhibitor will also inhibit this cell death, but the FAT and TRAD and RIP1 and RIP3 will bind with the caspase 8 will lead to combine with the RIP1 stimulation to the MLK combine and the RIP3, which that is the trimeric protein will lead to activate, and it will be bind with the mitochondria through a P-GAM-SL phosphorylation and P-GAM5S phosphorylation. And in this way here is the SER 637 will be activate with the DRPL to activate, and in this way bind with the mitochondria will lead to fission and necrosis. After this, here is the apoptosis. Let's begin to understand the extrinsic pathway first, and we will discuss next the intrinsic pathway. So the fat will combine with the receptor, the death receptor, the trail and FOS. The trail and fast receptor will bind and the procaspase 8 and procaspase 10, which that will be activated to caspase 8 and 10 and the bid activation. In this way, the bid activation will lead to the T bid. On the other hand, the caspase 3 and 7 activation via 9 and 8. So in this way, remember the 3 and 7 will lead to cause apoptosis. But on the other hand, we will also learn about the bid, and we will also understand about the intrinsic lethal stimuli like DNA damage, endoplasmic reticulum stress, and hypoxia. Hypoxia, which that will lead to stimulate the BH3 only protein, which that will basically stimulate the activated backs and back. BH3 only protein, which that will lead to activated the backs and back. This protein will target to the mitochondria for releasing of the cytochrome C. So after this is the mitochondria. When the backs and back will bind with the outer membrane of the mitochondria will produce the pores, and these pores will lead to the channel formation, will lead to the outflux of the cytochrome C. So the cytochrome C is the part of the mitochondria. Remember that cytochrome C is the part of the inner membrane of the mitochondria, and in this way it will release. After the releasing the cytochrome C and the APF1, the oligomerization will lead to combine together to form a apoptosome. This apoptosome is the complex formation due to the cytochrome C and the oligomerization of APF1, which that's combined together to stimulate the caspase 9. So remember, this caspase 9 will again trigger the caspase 3 and 7 to again the apoptosis through intrinsic pathway due to the damage of the DNA, endoplasmic reticulum damage or stress, and hypoxia, the low level of oxygen. While on the other hand, here is the XIAP, which that is stimulated by the SMAC. After the stimulation will lead to inhibit the caspase 9 for the inhibition of the apoptosis. While on the other hand, here is the BCL2 and XL and MCL1, which that inhibit the activated backs and back will inhibit the apoptosis. So let's begin to understand the necroptosis. The necroptosis is the different receptor involved. Here we will discuss tumor necrotic factor gamma receptor, T cell receptor, toll-like receptor 3 and 4, fast receptor, and trail receptor is the related apoptosis, including the ligand, and the tumor necrotic factor alpha receptor, which that bind this ligand, that several ligand, the specific ligand bind with this receptor. For example, the interferon gamma and CD328 and double-stranded RNA are lipopolysaccharide and trail you can understand. In this way, here is for example we are giving the example the TRAD and the TRAF 2 clap 1 and 2 and RIP 1 will combine together to tetramerically, and the clap 1 and 2 is the cellular inhibitor apoptosis protein, which that is the cellular inhibitor apoptosis protein. And in this way here you can see the ubiquitination will lead to separate the ubiquinone, and the CYLD will separate lead to used to RIP1 and FAT and caspase 8 activation, the trimerically lead to apoptosis. So this is the caspase inhibition, which that is basically the caspase inhibition will also occur. And in this way remember that things, the RIP1 and the RIP2, which that's combined together after the phosphorylation through caspase 8, and the basically it will lead to necrosome formation. This necrosome is containing RIP1 3 MLK, and phosphorylation will lead to necrosome formation here, and this necroptosis will be occurred by the binding of trimerically MLK with the phosphorylation lead to necrosis. But the inhibition through NEC1 glycogen synthase kinase 843 and 872 and NSA, which that will be inhibitor for the necroptosis. While the pyroptosis is a different, in this way the nanoparticle, the pro-inflammatory, will be endocytosed after the infolding of the plasma membrane of the cell. After the transfer to lysosome, this lysosome, when this vesicle will enter, is known as the endocytosis, and the transfer to lysosome. This lysosome is basically combined together to release this due to the pH increase, and in this way you can see induction of the pyroptosis will lead to inflammasome activation to the procaspase will convert into the caspase activation. And in this way here you can see the ACS is bound with the procaspase will release. After this here is a pro interleukin 18 and 1 beta, and the will activate the interleukin 18 and 1 beta. So remember that thing. And in this way here is the activation of caspases. The caspases activation will lead to caspases will lead to pro interleukin 18 and 1 beta will activate to interleukin 18 and 1 beta, which that will be separate. And in this way here is the GSDM cleavage also occur. Due to the GSDM cleavage will lead to give the binding of the membrane insertion into the plasma membrane, and in this way the creation of channel will lead to move water inside, while the interleukin 18 and 1 beta will move out, will lead to recruit other immune cell, while the swelling of the cell will lead to cell burst due to the increase of the water. So thanks for